Swayam Prabha. Digital India. Educated India.
Namaste. Moving on in our ecological structure module, we now move to the second lecture, which is species abundance and composition or biodiversity. Now, the ecological structure is comprised of a number of species, and so it becomes very important to understand how many species are there, how are they distributed, and what is the composition in a particular ecosystem. So, how many individuals are there of each species, and which is the study of biodiversity. Now, a number of ecological studies have been done in the forests, because forests provide an avenue in which you can understand nature in its most raw form, because they are untouched by humans. So, we are able to see how nature operates without any anthropogenic influence. So, if you enter into a forest, what do you observe? The first thing you will observe is a lot of serenity. You will not have any loud noises of vehicles, you will not have any smoke and so on, and you will also observe a number of trees. Probably if you are moving into forest, that is say a deciduous forest or a coniferous forest, so you will also observe a number of trees. Now, these trees will also be divided into a number of canopies. So, you will have a top canopy which is comprised of these tall trees, you will have a middle canopy, you will also have some ground cover that is comprised of these herbs and shrubs, and so on. If you look around and if you are lucky, you will also observe a number of animals in the forest. So, these are cheetahs which are feeding on the grasses that are found in the forest. So, you will observe some animals. You will also observe some interactions between these animals. So, like we talked before about the langur cheetal association. So, this is an example of a langur cheetal association. So, we have this tree on which we have this langur, and this langur is feeding and it is throwing off some pieces of the leaves down into the forest floor, and so there are a number of cheetals that have come to this area to feed on these leaves. Now, these cheetals would not have had access to the leaves because they cannot climb the trees evidently, and by coming in close contact with another species, now they are able to get an access to this food resource, and they are also benefited because the langurs can look very far because they are at a higher vantage position, and so if there is any predator nearby, if there is a tiger nearby, they would give off alarm calls and the cheetals would run away. Similarly, the cheetals are having another more ground view of the situation, and so if a tiger is nearby and if the langurs are not able to see it because of say tall grasses, the cheetals might be able to sense the presence of the tiger because of the smell of the tiger. If that happens, the cheetals would give off alarm calls and then they would start running off, and that would also alarm the langurs. So, we observe a number of interactions that are taking place between different species when we move inside a forest area. We will also observe a number of population level interactions amongst a number of animals. So, in this case we are observing a troop of monkeys that are doing allogrooming. Allogrooming is grooming of someone else. So, we will observe monkeys and their behaviors. If we look up, we will observe some birds. We will see parakeets most probably, or maybe even things like maynas or peacocks if we are lucky, or we will also observe some animals and birds that are migratory. So, this is an example of demersal cranes, which are migratory species that visit our country for a while and then they move off. We might observe some signs of some animals. So, for instance, this is an image of a pug mark of a tiger. A pug mark is a mark that is left on the ground when the animal is walking. So, for instance, in this case we have this loose soil. So, when the tiger was walking here, so it left an imprint on the ground which is the pug mark, and we have kept this pen to give you a sense of scale of how large this pug mark is. So, even though we are not able to see some animals, we might be able to infer their presence in our forest by using these indirect signs. Another indirect sign is the scat of animals. So, a scat is basically the fecal matter that is left behind by the animal, and every animal has a scat of different size, and they also have different behaviors when it comes to leaving their fecal matter. So, for instance, members of the cat family would typically after they are done disposing of their fecal matter, they would just move like this. So, they will scratch the ground. So, we will see some scratch marks near the scats, and we can make a correlation about the size of the scat, the number of scats or fecal samples that have been deposited by the animal, and some other signs such as scratching marks on the ground to infer which animal gave off this scat. So, we can also get a sense of what animals are nearby by looking at these grounds. Then if we look closely into the vegetation, we might also observe some reptiles such as chameleons or some other members of the lizard family. This area might also have some flowers and maybe some insects that are pollinating in this area. So, for instance, here we have a bee that is pollinating, and if you look closely, we will also observe some other body parts of the bee. So, in this case, these are the pollen sacs in which it stores the pollen. Then we might observe some other inferences such as termite mounds. So, if there is a termite mound, so you also have termites in this area, or things like fungi. Now, fungi play a very important role in the forest ecosystem, because when there is any dead wood or there are dead leaves that are lying on the ground, so they are also storing a number of nutrients inside. So, this dead log has some nitrogen inside, it has some phosphorus inside, it has some potassium inside, maybe some amounts of magnesium or iron as well, and by decomposing these logs, the fungi are able to release these nutrients back into the ecosystem, so that they can be made use of by some other organism. So, we will also observe some saprophytic organisms such as the fungi. There could also be a number of bacteria in this area, or maybe some other fungi that we are not able to look directly from our eyes, but we can make an inference. If we go near a water body, we might observe some reptiles such as mugger or some birds that will be seen near this area. In these water bodies, there will be some fishes, maybe some frogs, maybe some turtles. So, we might be able to see some turtles, or maybe we might not be able to see some turtles, and there will also be a number of plant life forms that are found in this area. And if we are lucky, we will even see a tiger somewhere, or some bears, or even maybe some elephants. Now, the point is, all of these are signs of biodiversity in this area, whether it is the tiger, whether it is the elephant, the plants, the trees, the bacteria, the fungi, all of these are signs of biodiversity. So, how do we define biodiversity? Biodiversity is the variety of life in all its forms and at all levels of organization. Now, in the last lecture, we looked at the different levels of organization that are found in the nature, and biodiversity refers to the variety of life. So, variety of life is different kinds of life forms that we are seeing in all its forms and at different levels of organization. So, when we say in all its forms, it includes plants, vertebrates, invertebrates, fungi, bacteria and other microorganisms. And when we say at all levels of organization, we can say diversity at the levels of genes, diversity at the level of species, diversity at the level of ecosystems, or we might even look at diversity at some other level of organization. Now, of these three are considered to be the most important: the genetic level biodiversity, the species level biodiversity and the ecosystem level biodiversity, and we will have a look at it in greater detail. So, what do we mean by now? Species, as we know, are groups of actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. Now, when we say actually interbreeding, so for instance, if we consider the cheetahs of Kana, they will be interbreeding amongst each other. So, they are actually interbreeding natural population. But if we consider the cheetals of Kanha and the cheetals of Rajaji, so both of these are not interbreeding with each other. Why? Because one is there in Madhya Pradesh and the other population is there in Uttarakhand, but they are potentially interbreeding natural populations. Why potentially interbreeding? Because say if you take a cheetal from Rajaji and take it to Kanha and try to mate it with the cheetals of Kanha, they will result in fertile offsprings. So, they are potentially interbreeding and they are natural populations. So, species are groups of actually or potentially interbreeding natural populations, and these groups are reproductively isolated from other such groups. What do we mean by reproductive isolation? So, for instance, you have a species that is called cheetal, you have another species that is called say tiger. If you try to mate a cheetal with a tiger, they would not be able to mate, or even if in extreme situations if we are able to coax them to mate, they might not result in an offspring, or they might even result in an offspring that is itself infertile. So, this is meant by reproductive isolation. So, they are reproductively isolated from other such groups. So, species are groups of actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. Now, species biodiversity asks how many species are there and how are they distributed. So, the number and the distribution. The genetic biodiversity on the other hand asks the diversity of genetic information that is present at the level of phyla, families, species, populations and individuals. Now, genes as we know are units of heredity that are transmitted from parents to offsprings. So, we might have a gene for say eye color, a gene for hair color, a gene for skin color, a gene for tallness and so on. So, when we consider all these different genes, genetic biodiversity asks what is the diversity of genetic information. So, for instance, if you consider the diversity of genetic information at the level of a population. So, for instance, consider a population of cheetals in Kanha. So, how different are these cheetals from each other genetically? Are all these cheetals having the same gene for height, or are they having different genes for heights? Are they having or different alleles for the height, or are they having the same coat color, or are they having the same eye color and so on. Now, examples of genetic biodiversity include polymorphism and heterozygosity. Now, polymorphism is the proportion or percentage of genes that are polymorphic. Now, a gene is considered polymorphic if the frequency of the most common allele is less than some arbitrary threshold, and this threshold is generally taken to be 95%. Now, what do we mean by this? Now, let us consider the coat color. The coat color of cheetal. So, let us say that it is coded by a gene called C. Now, this gene C might be available in a number of forms. So, we might have a C1 allele that codes for a very light coat color, and we might be having say C2, C3, C4 and C5, and C5 codes for a very dark coat color, and these three are coding for some intermediate coat colors. Now, any particular organism or any particular individual of cheetal in this population will be having two alleles for the same gene C. So, basically it will be having two C's, one is coming from its father and the second one is coming from its mother. Now, it is possible that you have C1 and C1, C1 coming from father and C1 coming from mother, or you could have C1 and C5, or say C3 and C5 and so on. So, if you consider all the individuals in this population and you figure out what alleles are there, and then you do a counting of these alleles. So, it is possible that we might see that C1 is present 200 times, C2 is present 100 times, C3 is present 150 times, C4 is present 1000 times, and C5 is present say 10,000 times. Now, in the case of polymorphism, we will ask the question, is this gene, the gene for coat color that is represented by C, it is polymorphic if the frequency of the most common allele is less than 95%. Now, in this particular example, how many alleles do we have? Number of alleles is 200 plus 1 300, 450, 1450, 1100 and 450. So, these are the total number of alleles that we have. Now, what is the frequency of the most common allele? Here the most common allele is C5, which is present in 10,000 copies. So, the frequency is given by 10,000 divided by 11,450 into 100. Now, this is the frequency of the most common allele. Now, the question is it more than 95% or is it less than 95%? So, if we do this calculation, 10,000 divided by 11,450 multiplied by 100, it comes to 87.34%. Now, is this less than 95%? The answer is yes. So, in this case we would say that this particular gene is polymorphic for this particular population. Now, suppose we had, say, in place of 10,000 genes we had it in 100,000 genes. Now, what would be the total in this case? So, we have 1450, 101450, and in this second example what would be the proportion of C5? So, here the proportion of C5 would be given by 100,000 divided by 101450 into 100%. Now, if you do this calculation, so we have 100,000 divided by 101450 into 100 will get to a figure of 98.57%, which then would be greater than 95%. So, in such a scenario we would say that this particular gene of coat color represented by C is monomorphic for this particular population. So, essentially what we are asking is that if there is a gene that has only one allele, so for instance if all these individuals were one and the same with regard to coat color, everybody just had C1 C1. So, in that case this particular gene would be called monomorphic. Mono is one, morpho is form. So, it has only one form which is C1, but if we have more than one forms we then put a threshold. Is this threshold so when we have this threshold of 95%, we ask if the frequency of the most common allele is greater than this or less than this, because for instance if you have a population in which you have 1 lakh copies of say C1 and say only 2 copies of C2. So, in that particular case we would say that even though we have 2 different alleles for this gene, but it is still monomorphic, because more or less we can say that all the individuals are similar when it comes to the coat color. Now, the second thing that we ask in the case of genetic biodiversity is the level of heterozygosity. Now, heterozygosity is the proportion or percentage of genes at which the average individual is heterozygous. So, what we ask in this case is, suppose in the case of cheetal we have 100 genes. Actually this number of genes is very large, it is say between 20,000 to 100,000, but for an example let us take 100 genes. Now, heterozygosity asks the question, what is the percentage of genes at which the average individual is heterozygous? So, heterozygous means that suppose for gene 1, this gene was present in 2, every gene is present in 2 copies. So, suppose this was present as 1 1 and 1 1. So, in this case we would call it homozygous for gene 1. Now, in the case of gene 2, suppose it was present as 2,1 and 2,4. So, here we will say that it is heterozygous for gene 2, and similarly we will move on till the last gene, which is the 100th gene. Now, in this case, suppose out of these 100 genes there were 40 genes for which this individual was heterozygous. So, in this case 40 out of 100 genes it was heterozygous. Now, we take another individual. For this individual it was say 35 out of 100, maybe for another individual it was 70 out of 100 for which it was heterozygous. Now, heterozygosity would ask what is the proportion or percentage of genes for which the average individual is heterozygous. So, we can average out all of these values to get an idea of the level of heterozygosity that is present in this population. So, genetic biodiversity can be looked at using these two examples, polymorphism and heterozygosity. Next is ecosystem biodiversity. Now, ecosystem, as we saw before, is a group of interacting organisms or population or community and the physical environment they inhabit at a given point in time. So, we have a group of different populations that forms a community, and to this community you add the abiotic elements or the physical environment and you get the ecosystem. Now, ecosystem biodiversity asks the question, how many ecosystems are there and how are they distributed? Now, what are the different kinds of ecosystems that we can have in a forest? So, suppose this is our forest. Now, it is possible that the whole of this forest is comprised of trees. So, in which case we will say that it has only one kind of an ecosystem, but then it is also possible that this forest has this area that is a wetland, then it has this area which is a grassland, then this has this area which is a rocky outcrop, then probably you have this area which is very close to a river, and so it gives you a different kind of an ecosystem, and rest of the area is comprised of trees. Now, in this case we would say that this area is comprising of a forest ecosystem, a grassland ecosystem, a wetland ecosystem, a rocky outcrop ecosystem and a riverine ecosystem. So, in this case we have 5 different ecosystems. Now, ecosystem biodiversity is asking the question, how many ecosystems are there? In the first example we had only one ecosystem. So, in the first example we only had this forest and the whole of the forest was comprised of trees. So, this is one ecosystem and this is another ecosystem. So, we can see very clearly that this ecosystem with 5 different smaller ecosystems is much more diverse as compared to the first case in which we have only one ecosystem in this area. The second thing that it asks is how are they distributed? Now, why is distribution important? Distribution is important because, say in this particular example, in place of having a scenario like this, we had a rocky outcrop that moved like this. Now, so this is a rocky outcrop. Now, what is the difference that we get? Now, in the first example, we had this rocky outcrop and here we had the water body. Now, in this case, let us look at the number of edges that we have in this small portion only. So, we have this edge that is comprised of rocky plus forest, and we have this edge that is comprised of wetland plus forest. But so let us call this situation 1 and let us call this situation 2. In this situation we have an edge which is rocky plus forest. We have this edge which is wetland plus forest, but now we have another edge which is this one, which is rocky plus wetland. Now, by having such a distribution, by having more number of edges, we also increase the amount of biodiversity in this area. Why? Because there might be some species that prefer living on the edges. So, for instance, there could be a species that prefers living here, so that it is able to venture into the forest so that it can get its food, but when the predators come, it should be able to run into the rocky outcrop to save itself. So, edges have their own significance in the ecosystem. Now, by having a distribution in which maximizes the edges that are possible, we can maximize the biodiversity, which is why the ecosystem biodiversity asks this question, not only how many ecosystems are there, but also how are they distributed. Now, out of these different approaches of measuring biodiversity, the simplest one is looking at the species richness and the species evenness, which is the species biodiversity. Why? Because in the case of genetic biodiversity, you have to look at individual genes that are present in the population, which is not only very technology intensive, but also very much cost intensive. But if you just went into the forest and looked at the number of species that are there, so they can just be looked with your naked eyes, or maybe with using a binocular or some microscope. So, it is the most easiest way of measuring biodiversity, and so this is the most widely used measurement of biodiversity. Now, species biodiversity also asks two questions. One, what is the number of species that are present in your area, and two, what is the distribution of individuals of different species. Now, what do we mean by that? The first one is simple. Species richness is the number of species present in an area. So, consider two forests. So, you have this forest 1 and you have forest 2. Now, say forest 1 has a total of 100 species and forest 2 has say 1000 species. Now, species richness asks the question, how many species are present. So, in this case you have 1000 species, here you have 100 species. So, we will say that forest 2 is much more biodiverse as compared to forest 1. So, that is species richness. The second thing is species evenness, or the distribution of individuals of different species. So, for instance, in this case, suppose both the forests had equal number of species. So, let us have 100 species in forest 1 and 100 species in forest 2. Now, the distribution of individuals of different species asks the question, how many individuals do we have of each species in both of these forests. So, in this case, suppose species 1 has 10 individuals, species 2 has 12 individuals, species 3 has say 15 individuals, and so on till species 100 that also has say 20 individuals. So, this is the level of distribution of individuals of different species that we have in forest 1. In the case of forest 2, suppose we have species 1 that has 2 individuals, species 2 has 3 individuals, species 3 has 2 individuals, and so on, but there is one species that has say 10,000 individuals. Now, in this case we would say that even though both the forests have the same number of species, both have 100 species, but if we look at forest 1, so in this case all these species have roughly equal number of individuals. So, when you move out into the forest, if you take any snapshot, you might be able to see all 100 of these species. So, all these 100 species are distributed everywhere, whereas in the case of forest 2, we have 99 species with very few number of individuals, and one particular species that has 10,000 individuals. Now, if you move into this forest, you would observe only species number 100, because rest of the species are so few that that your system is overwhelmed with species number 100. So, in this case we would say that the species are not equally distributed, or the individuals are not equally distributed amongst different species. So, when we ask this question, what is the level of species evenness, distribution of individuals of different species, we would say that even though the species richness is same in both these forests, 100 species each, but this forest is much more even and this forest is very much uneven when it comes to evenness. So, when we have the same level of species richness, we prefer an even forest. So, we will say that the amount of biodiversity in forest 1 is much greater than the amount of biodiversity in forest 2. Now, how do we know how many individuals are there in the forest? So, that brings us to a concept of species accumulation curve. So, how do you get to know how many species are there in a forest? Let us consider that we are considering the number of mammals that are found in a forest. So, we are saying the number of mammals in the forest. So, you go into the forest and on the very first day, suppose you saw 10 species, this is 10 species. So, you have the number of species and you have days, and on every day, suppose you are venturing out into the forest and you are spending say 6 hours in the forest and you are looking at different mammals that you can see in this forest. So, on the very first day you will be able to see the maximum number of animals, because these are the most common animals. So, you venture out into the forest and you saw say cheetal or sambar or langur or maybe some macaques. So, these are the animals that are very easily seen. Probably on day 2 you did not find any new animal, so the number of species remains the same. So, this is day 1, this is day 2. On day 3 you got a bit more lucky and you saw a tiger. So, on day 3 your number of species increased from 10 to 11 because you saw a tiger. On day 4 it is possible that you also saw 2 other animals. So, it increased from 11 to 13, maybe because you saw a leopard and you also saw an elephant. But then with time, as you are observing more and more number of species, it will reach to a point of saturation. So, when we draw and the number of days and the number of hours that are spent goes by the name of effort. So, we are drawing the number of species versus the effort. So, on the very first day you will see the largest number of species, then this curve would increase with time. It might also remain constant for some days, but then it will again increase as you are going to see more and more number of individuals. But then after a while it will become flat, because for so many days you are not seeing any new individual. So, here we see that on the first day we saw so many individuals, then every day we are seeing some new individuals, but then after a while it is reaching to this level of flatness. And once we reach this stage, we say that this is the number of species that are found in this area. So, we draw a straight line and we see that this is the number of species that we have in this area. Of course, the number of species may be a bit more or a bit less, but more or less this is the number of species that we have. So, this is a way in which we get to the species richness of an area. Now, after richness, the next thing is to compute the species evenness. So, for all of these species, we will keep a note of how many individuals did we observe. for this particular effort, and then we would compute it in this way. So, say you have the ith species and you have the population of the ith species. So, 1, 2, 3, 4 and so on. So, suppose for the first species you saw 100 animals, for the second species you only saw 2 animals, for species 3 you saw 30 animals and so on. So, you will make this table, and once that is made, we measure biodiversity using different indices. Now, the first index goes by the name of Simpson's diversity index and is given by the symbol D, which is equal to 1 over sum I is equal to 1 to S and pi square. Now, this let us call it as small pi, and from this we will compute a capital Pi. Now, here we have total number of species. So, S is the last species that we have. Let us say we have 100 species that are found in this area, and the total number of individuals that we found in this area is 1000. So, this is the sum of all the PIs is 1000. Now, capital Pi is computed as small pi divided by the sum of small pi's. So, in this case, because this is 100, so you will have 100 divided by 1000 is equal to 0.1. In this case, you will have 2 divided by 1000 is 0.002. Here you will have 30 divided by 1000 is 0.03 and so on. So, here we are computing the PIs. Now, in the case of the Simpson diversity index, you have 1 divided by the sum of pi square. So, in this case, you will have pi square. So, let us draw it again. So, here you had pi. So, pi was 0.1, 0.002 and 0.03 and so on. So, you do a pi square and you get 0.01, and then this will be 4, 1, 2, 3, 4, 5. This will be 9 and so on. So, here you are having the pi squares. Now, you do a sum of all the pi squares and you will reach to a value of x, and then D is given by 1 divided by the sum of all the pi square or the sum of or this value of x. So, this is the measure of biodiversity that is given by the Simpson's diversity index. Now, D is the Simpson's diversity index, S is the total number of species in the area, and pi is the proportion of the ith species in this area. Now, from this D we get to a value of evenness. Now, evenness or E is given by D divided by the maximum value of D or D max. Now, evenness is maximum if for all of these species you had the same number of individuals, or best if you had only one individual of all the species. So, for instance, in this case species 1 had only one individual, species 2 had only one individual, species 3 had only one individual and so on till species number 100. If that is the situation, so mathematically the value of D will become maximum, and when you divide D by the maximum value of D, you get the equitability or evenness value of the Simpson's diversity index. Another index that is used is the Shannon diversity index. Now, in the case of Shannon diversity index, you have its minus sum over I from 1 to the last number of species, that is I is equal to 1 to S, and here you have pi log of pi, and this log is the natural logarithm which is log to base e. So, once you compute this value, this is the Shannon diversity index, and here also you can get to an evenness value by h divided by h max. Now, these formulae do not have to be remembered for the purpose of this course. This is just to give you an indication of how this biodiversity is actually measured in the field. Now, from here we move to the next concepts of biodiversity. We have geographically we can have three values of biodiversity: alpha, beta and gamma. Now, alpha biodiversity is the biodiversity that exists within an ecosystem. So, in our forest, so this is our forest, and in this forest we are having 5 different ecosystems. So, the first ecosystem is the forest, the second is the rocks, the second is the grasslands, the third is the wetland, fourth is the rocks, and fifth one is the riverine ecosystem. Now, alpha biodiversity will ask the diversity that exists within an ecosystem, or for instance, if you are looking at alpha biodiversity in terms of species richness, how many number of species do we have in the forest ecosystem or the trees ecosystem? How many number of species do we have in the grassland ecosystem? So, suppose here you have the number of species is 20, here you have the number of species is 10. So, alpha biodiversity is asking the question, what is the biodiversity that exists within each and every ecosystem? Beta diversity on the other hand asks what is the diversity that exists among different ecosystems. So, for instance, it is possible that out of these 10 and 20 species, there are say m number of species, say 5 species that are common between both of these. So, there are only p number of species, that is here you have 15 and here you have 5 species that are different between both of these ecosystems. So, beta diversity asks the question, what is the diversity that exists among different ecosystems? And gamma biodiversity takes it even further to ask what is the diversity that exists among different geographies which are very far from each other. Now, to give an example of alpha, beta and gamma biodiversity, here is a field situation. So, you have 2 islands. On the first island you have a patch of trees and a swampy area, and on the second island you have a patch of trees. Now, here we are looking only at the lizard population that is there. So, this swampy area has one species of lizard, this patch of trees has got 2 species of lizards, one lives on the grasses and the second one lives on the trees, and on this island we have another species of lizard that is found. Now, when we ask about the alpha biodiversity, here we have two species of lizards, here we have 1 species of lizard, and here we have 1 species of lizard, which is the alpha biodiversity. Now, if for instance we try to increase the alpha biodiversity of this particular ecosystem or this particular island by draining off the swamp and generating a piece of woodland in this area. So, in that case this species would be lost, but we would be getting 2 species in the same area. So, in this case the alpha biodiversity would increase from 1 to 2, because now we only have 1 species, and if you drain out this swamp and do a copy paste of this area, you will get 2 species. But if you look at the beta biodiversity, so currently between these 2 ecosystems we are having 3 species, 2 here and 1 here, but when we replace this swamp with this forest land, we will only be left with 2 species. Or to put it as an image, in the first case we have these trees and we have the grasses, and on the other hand we have some swamps. In the second case we would drain off the swamp and we would have trees here and trees here, and we would have grasses here and we would have grasses here. But in the first case we had a species 1 here, a species 2 here and a species 3 here. In this case we would have species 1 here and species 1 here, species 2 here and species 2 here. So, if we look at the alpha biodiversity, so here alpha is 1, here alpha is 2, because for this particular region, the swampy region, you have shifted from one species to two species. But if we look at the beta biodiversity, here beta is 3, whereas here beta is only 2, because here you only have 2 species that are there on this island when we are looking at the beta biodiversity. So, it is important to look at biodiversity at different levels of organization. Now, we make use of all this information to decide where to concentrate our conservation efforts. So, we define some areas as biodiversity hotspots. Now, biodiversity hotspots are areas with high species richness. So, they have a large number of species, a high degree of endemism. So, endemism means that the species that are found in this area are not found anywhere else. So, these areas are providing a very typical ecosystem or a very distinctive ecosystem for the species that are found here. So, because this distinctive ecosystem is not found anywhere else, so this area has a very high degree of endemism and also a very high degree of threat. So, a high degree of threat would mean, for instance, if we have a new disease that is taking over the individuals of this area. So, for instance, in the case of South America, we have a fungal disease called the chytrid disease that is exterminating the populations of frogs. So, a number of species of frogs from that area are getting extinct. So, we will say that those spots in South America have a very high degree of threat. Or for instance, let us consider the equatorial rainforests. Now, a number of equatorial rainforests are being cut off to gather timber, or maybe to create some settlements, or maybe to construct some roads, or to have minerals by converting those areas into mining areas. So, there again, because these ecosystems are getting destroyed, we will say that they have a high degree of threat. Now, biodiversity hotspots will be areas that have all three of these. So, they have a large number of species. These areas are providing such ecosystems that these species thrive only in these areas, they are not found anywhere else. So, they have a high degree of endemism and they also have a high degree of threat. So, once we do that, we will observe that there are certain areas that require a larger degree of protection as compared to some other areas. So, for instance, in the case of India, if you look at the Western Ghats, so Western Ghats are a biodiversity hotspot. Why? Because they have a very large number of species. These species are not found anywhere else. So, for instance, some frog species that have been discovered in the Silent Valley National Park of Kerala are not found anywhere else in the world. So, those species would be said to be extremely endemic to those areas, and these areas are also having a large amount of threat because people want to chop off the trees here and convert these areas into plantations, because these areas are also very good areas for say coffee plantations or for say rubber plantations. So, in this case, these areas would be classified as biodiversity hotspots and we would need to focus more attention into these areas. Now, in this lecture we had a look at the ecological structure in terms of biodiversity. So, we looked at species abundance and species distribution and species composition of different areas. So, we defined biodiversity as the amount of variety that exists in different life forms, in different forms and in different levels of organization. We defined genetic biodiversity, we looked at species biodiversity, we looked at ecosystem level biodiversity, and we also looked at alpha, beta and gamma biodiversity, which is biodiversity within an ecosystem, biodiversity between two ecosystems and biodiversity between two different geographies. And we also had a look at how to determine the species richness and the species evenness of an area by looking at the species accumulation curve and also by looking at the number of individuals that are present for every species. And to concentrate all this information into one index, we can develop we can make use of the Shannon index or the Simpson index, which gives us a snapshot view of the amount of biodiversity that is present in an area in terms of the species richness and in terms of the species evenness. And from that we moved on to the biodiversity hotspots, which are areas which have a very large number of species or a large species richness. They have a high amount of endemism, which means that those species are only found in those areas, they are not found anywhere else. So, if we lose those areas, we will lose those species forever. And 3, these areas also have a high degree of threat, because people want to divert these areas for some other applications, or probably because these areas are suffering from some diseases, or maybe they are suffering from heavy floods, or maybe forest fires, and so on. Now, these areas which are defined to be biodiversity hotspots are those areas whose ecosystems require a larger degree of protection if we wanted to conserve the wildlife in this area. So, that is all for today. Thank you for your attention. Jai Hind.